In order to understand how skeletal muscles contract, we have to know about action potentials and calcium. We last left off learning about how sodium ions enter into the skeletal muscle fiber at the neuromuscular junction. So we're going to pick up from there. That is eventually going to cause something called an action potential. So the acetylcholine opens those ion channels, and that leads to a traveling change in voltage called the action potential. This traveling change in voltage occurs along the cell membrane, along the sarcolemma. This action potential is going to act as a signal in the muscle fiber, and this signal will cause the release of calcium from the sarcoplasmic reticulum. Calcium will then lead to the events that allow myosin to grab onto actin and pull, which is the actual contraction part. So let's take a look at that action potential. Acetylcholine is that neurotransmitter released by the neuron at the neuromuscular junction. The acetylcholine travels across the cleft and binds to receptors in the cell membrane of the sarcolemma. We call that area of the sarcolemma the motor end plate. The acetylcholine would open the receptor because the receptor is a chemically gated ion channel. Acetylcholine is the chemical that opens the gate, and when the gate is open, ions, specifically sodium, can flow through. And because there's a greater concentration of sodium outside of the muscle fiber, the sodium goes from the area of high concentration outside to the area of low concentration inside. So we have all of this sodium rushing into the muscle fiber, and sodium has a positive charge. And that's going to add positiveness to the inside of the muscle fiber. And that's going to change the voltage inside the muscle fiber. When the inside of a cell becomes more positive, we have a special term for that called depolarization. And we'll learn more about that when we get to our neurophysiology unit. But the entry of the sodium causes that depolarization, that increase in voltage inside the muscle fiber. A muscle fiber is typically at a resting voltage of around negative 90 millivolts. Believe it or not, we can put little voltmeters inside and outside of cells and measure the voltage difference between the inside and the outside. And when we do that for a skeletal muscle fiber, we see the difference in voltage between the inside and the outside is 90 millivolts. And we say it's negative 90 millivolts because the inside is more negative than the outside. Once that sodium comes in, we're not going to be at negative 90 anymore. We're adding positiveness. So we might go up to negative 80, negative 70, and eventually maybe reach to like negative 55 millivolts. This depolarization, this increase in voltage, is important because it's going to open voltage-gated ion channels. Right? Acetylcholine opens a chemically gated ion channel, but this change in voltage from the sodium coming in is going to open another type of channel embedded in the cell membrane in the sarcolemma of the muscle fiber. And it's going to open these voltage-gated sodium channels. And when we open one voltage-gated sodium channel, we're going to open them all because they kind of work like dominoes. You knock the first one down, they all fall down. You open one voltage-gated sodium channel, they all open. And you know how that wave of dominoes falling down occurs? Well, we're going to get a wave of opening voltage channels, and that's traveling, that's a movement, and that's our traveling change in voltage that is the action potential. To get a better idea of that process of depolarization, let's take a look at this little animation. So the red pluses are sodium ions. We'll call the area above the cell membrane outside and the area below the cell membrane inside. There's a lot more sodium outside the cell than inside. As sodium rushes into the muscle fiber, it's going to cause a local depolarization, a local change in voltage. So let's put this together with what we already know. The acetylcholine will bind to the receptor, opening it, allowing sodium to come in, and now we get a depolarization because that positive sodium coming in means we're adding that positiveness to the inside. So in this example, we went up to negative 55. That's a depolarization because negative 55 is more positive than negative 90. When the local area depolarizes to negative 55, nearby neighboring voltage-gated ion channels are going to open one after another all along the sarcolemma like dominoes falling down. And that is the action potential. So let's add that into what we already know. Again, the top is outside and the bottom is inside. Lots of sodium out, not very much in. Acetylcholine comes along and it binds to the acetylcholine receptor. This is the chemical that opens the gate. So now the gate is open. This is a chemically gated ion channel. So ions are going to now flow through the channel, specifically sodium ions. And the sodium ions go from the area of high concentration, outside, to the area of low concentration, inside. And as the sodium comes in, it adds that positiveness to the inside, that depolarization, and the voltage changes to about -55 millivolts. Notice the plus/minus here. This is not a chemically gated ion channel. This is a voltage gated ion channel. And this -55 is the change in voltage that we need to open it. Once we open that voltage gated ion channel, more sodium will come in, causing the next area to depolarize, and this will cause the next voltage gated ion channel to open, which would cause more sodium to come in, causing the next voltage gated ion channel to open, which would cause more sodium to come in, and you can see how we get that traveling change in voltage. That is the action potential.